some of the work that's going on currently in your institution?
Our institution is focused on understanding control of neural stem cells. Their existence is fairly modern, 15 or 20 years ago, and these are highly proliferative cells that can make various parts of the brain. In adulthood, these cells remain dormant, and in humans, they're very dormant and hardly repair injury after something untoward happens. In lower animals, they can repair. And so we look at the mechanisms involved in controlling their activation for regeneration and repair in the brain, in the spinal cord, and increasingly in the retina, which is part of the central nervous system.
Now, the blood-brain barrier, or rather the nerve, the retinal layer, has a number of very distinct properties that make it different from other interfaces in the body. Tell us about some of your work in characterizing that and what the implications are for the placement of stem cells.
Well, thinking about other places of the body to begin with, the bone marrow, we're born with 1 million stem cells that remain dormant throughout life and are activated for regeneration and repair of the blood system. Same in the skin, we have stem cells laying dormant that can be activated to repair a cut. The retina presents an extremely well-defined niche. It's a two-dimensional structure where we work particularly on the retinal pigment epithelial cells are sandwiched between the neural retina that converts light into vision and a dense membrane under that, and are adjacent to each other in a highly ordered structure that maintains their dormancy through life. And the retina, I think, presents a uniquely accessible niche for controlling these RPE stem cells. We can see it through the pupil, and we can image it in many ways and manipulate it in many ways.
So what are the approaches that you're looking at to the activation of dormant cells or the implantation of stem cells?
You know, the tools that are becoming available over the last few decades are remarkable. Molecular tools, we can now look at the RNA transcripts from a single cell and compare that to its neighbor, compare that to a cell that's been treated in such a way, anyway. And much of our work begins by removing these cells into tissue culture and observing them under a microscope and applying various factors that constitute the niche that controls their behavior. So we do time-lapse movies to watch their proliferative behavior and understand the lineages that are created as they divide to make various progeny. And antibodies are available to look at which molecules are turned on or off. And again, new technologies have evolved to understand the changes in RNA and DNA that occur during activation, even on a single cell level.
Tell us about the work that you've presented here at ARVO 2016.
The retinal pigment epithelium develops very early in humans. And so the cells, the dark cells they're called, the black cells in the middle of our pupil, haven't divided since very early in development, 6 weeks in human. Many of these cells have stopped dividing well before birth. When we take them out of a donated eye and culture them, there's a subpopulation that behave like early developmental cells and divide. There's stem cell population that reproduces itself. We've long known that RPE proliferates in a culture dish. What we're finding now is a subpopulation of cells, a few percent, self-replicate, make copies of themselves so we can expand to large numbers of cells. And these cells are poised, these progeny are poised to redifferentiate into RPE, genuine RPE. And so for the first time, I think stem cell technologies in general allow us to have many human cells available for study and for transplantation. And the work we presented at ARVO 2016 was aimed at transplanting the adult RP stem cell, derived RP progeny, into an animal model of macular degeneration. And the key result that was new for this meeting is that the RP stem cell-derived RP rescue vision in these animal models, and we found a specific stage of differentiation is more efficacious at rescue. We've long known that younger cells are more reparative, and others have found that after transplant, younger tissue or younger stem cell-derived progeny are more effective at repair. And we've now confirmed that is also the case for RPE.
When you say younger cells, you mean at an earlier stage of differentiation or genealogically younger?
Well, the definition of younger is an interesting one, and in this sense I mean developmental stage, how differentiated these cells are. And the epigenetic clock continues to tick, and in that sense these cells are quite old. They're from donors who may be 80 years old. And what we find is they behave after activation the same as cells, or very similarly to cells from younger donors, and there's a reset of the epigenetic clock.
In the animal models, did you implant them into the subretinal space? What is the actual location of the implantation?
The normal RPE is in the subretinal space, and we transplant directly to the subretinal space.
And what was the persistence of the cells like? How long did they survive?
It's an excellent question. A difficult one to answer in terms of the macular degeneration foundation, because the rodent models we're using only live for a few years, and we do see persistence up to 180 days, over 180 days now of vision rescue.
Were there any specific external variables that you found were predictive of success in this process? Apart from having the genealogically youngest donors and having technical success in implantation, were there any other factors associated with success? Nutritional factors, environmental factors, other cofactor illnesses? Did you induce metabolic syndrome in the rats?
The history for younger cells being more reparative and regenerative goes back hundreds of years. And our experiment was based on many other tissues. What we've done is look forward from here. These cells that are differentiated for 2 weeks or 4 weeks or 8 weeks have the same genotype. They're maintained under identical conditions. The only difference is 2 weeks. So we've been able to harness these new molecular technologies to find out what is different, what changes over those 2 weeks that confers efficacy at vision rescue. And we have now a list of molecules, one of which we're pursuing a long non-coding RNA to try to gain an early understanding of why younger cells are more reparative, at least for the RPE.
Now, there's obviously been a discussion of gene therapy alongside stem cell therapy. And are you pursuing this particular avenue, a combination for certain diseases that have a major genetic driver?
Some are. We are not. I think our perspective, comparing gene therapy and cell therapy, I'm going to bring in the engineering chips that are being implanted as well, which give a glimmer of vision that is invaluable to totally blind individuals. I think the gene therapy is most valuable at the other end of the spectrum. Early stages of monogenetic diseases, where we can correct or others can correct the defect to prevent the damage. And cells, regenerative therapy, fill in that middle zone where macular degeneration and many degenerative diseases of aging or trauma reside.
So tell us about the next steps now, based on your work that you've presented. What are the next things in the pipeline for you?
Well, the one that we're most excited about is based on the fact that our cells, the RPE stem cells, are already present in the eye, raising the question of whether they need to be removed for activation. We know the factors needed to activate them, and we are now developing controlled release materials that activate these cells in culture. And we've gone to look at explants of RPE and Bruch's membrane for macular degeneration patients and can activate the RPE in those aged human explants, and are moving further to inject the controlled release material, growth factor releasing material, into the eye of animal models and see activation there. So the hope is that since the cells are already present, we won't need to remove them. We can activate them in situ to gently stimulate self-repair, which, as we mentioned earlier, is present in younger animals or less evolved animals, but in the adult human inhibited. We believe the machinery for self-repair is present in this RPE stem cell, and we are attempting to activate it for endogenous self-repair without transplantation.
I think that's a very, very powerful model. And it's almost, one would say, the holy grail in this field. And you feel confident that you've identified molecules that have great promise in initiating this activation.
Well, we are activating this. We are activating the RPE stem cell to gently proliferate in situ. I'm glad to hear that this is the holy grail, because I often felt we are out in a limb, that everyone wants to transplant stem cells. And we are doing that, and that's our main work right now because it's most accepted. I think that the concept of activating dormant repair mechanisms that are already present is the future, and we're excited to pursue and participate in developing that technology.
It's physiologically very elegant. It's something that appeals to me as an internist, that it has both a physiological and biochemical elegance to it, which implantation doesn't quite do because of the interventional nature. So as you see your particular program panning out, what are your next milestones in this process?
Well, we're currently most actively pursuing the transplant approach, which I believe shows proof of principle that increasing the number of RP cells in animal models of AMD rescues their vision. And I think it's important to show proof of principle for increased number of RPE having efficacy. Transplant's a very simple way to do that. We are planning a phase 1 clinical study. We're still about a year and a half, probably 2, from our first patient. And immediately behind that, developing the controlled release mechanisms for activating the endogenous repair mechanism. Cause RP very gently to divide once or twice over a few-week period. Developing the material under GMP conditions that are required by the FDA for a clinical study is just beginning and is quite a lot of work. And those are our most concrete next steps.
And your work is supported by the NIH, the NEI?
Yes, we're supported mostly by grants. The New York State Stem Cell Board, we call NISTEM, has made progress accelerated for us very much.
Are you currently recruiting patients?
No. We are currently in the middle of writing an IND application, new drug application to the FDA. Again, another year and a half or so, and hoping that they allow us to move into patients soon after that.
What should patients take away from your current field of research? What are you currently doing, and what is the promise for patients, if you were to describe it in lay terms?
Degenerative diseases like AMD, I think stem cell therapies hold great hope and are the leading technology. Balancing that hope with the reality is a very important thing to understand. We're not ready to treat AMD now, unfortunately. We're making progress. The rate of that progress is unpredictable. We hope it goes smoothly and quickly. Follow progress of ourselves, and there's an American company, Okata, that is already into phase 2, transplanting embryonic stem cell derived RPE cells, and their clinical studies are reporting positive results. The hope is real. On the other hand, there are many charlatans coming on the back of this real work. Areas that are to clinics that are charging money for a therapy that is unproven is, in my opinion, not ethical. And education, to the patient, please follow progress and understand and ask your retina specialist or your ophthalmologist what is real and what's not, and understand the difference. The reason I'm here with you, David, is that I believe in that education very much. I'm grateful to you and the Macular Foundation for carrying out these videos to educate the patients about the hope that is rapidly heading their way.
Excellent. Professor, is there anything else that you'd like to add about your work and about other activities?
Well, maybe a general thought is an optimistic one, that the tools that are available, a point I made at the beginning of this interview, are remarkable right now for advancing biomedicine. And support our government, support the NIH or the New York State Stem Cell Board, urge the politicians to help these entities fund the research that's needed in order to deliver safe and effective new therapies. The potential over the next decade is remarkable. The occurrences over the past decade have been remarkable. And it's a very exciting time to be involved in research for benefit of macular degeneration patients.
Professor, thank you so much.
Thank you so much,
Great.
David. Thank you.
Absolutely excellent.